Hi guys, today we will be discussing Tryptophan Operon. Now you already know that you have studied about lac operon, and you have seen that in lac operon was first catabolic operon. It was first a catabolic operon. Why it is a catabolic operon? It is a catabolic operon because it can only be activated when there is a presence of lactose. If lactose is not there, then it will not be activated, and they are only activated in order to hydrolyze or degrade lactose. So it is a catabolic operon. Moreover, the repressors of your lac operon are actually synthesized only under certain conditions. So those are basically inducible repressors. That means they are being produced under certain conditions. They are not constitutive repressors that will always produce even if their condition is not favorable. So that was your catabolic operon. But as we discussed in this video will be based on your anabolic operon, and this anabolic operon basically comprises of trip. It is your trip or your tryptophan operon. It is your tryptophan operon. Now the most important thing in tryptophan operon, it is actually a anabolic operon. First point, it is a anabolic operon. Why it is called anabolic operon? It is called anabolic operon because it synthesizes tryptophan. That means this operon catalyzes the synthesis of tryptophan when there is deficiency of tryptophan in the cell. Tryptophan is a very essential amino acid which is need to be synthesized by the cell, and hence this operon is also present in the microorganism called Escherichia coli. Now let's move on to the genomic organization of your tryptophan, and you get to see that this is a genomic organization which I am drawing for tryptophan operon. Now this tryptophan operon basically comprises of R, which is your repressor, then it is having a promoter, then it is having a operator, and then it is having some genes which are, there is a sequence which is called as your leader sequence, which is basically called as your leader sequence, and then it is having 5 structural genes, and this 5 structural gene is your E, D, C, B, and A. So it is having 5 genes. So these are R, C, and O. R stands for repressor. So let's write it. R stands for repressor. So R is a repressor. C is your promoter. O is your operator. Right? So what are these? Repressor, promoter, operator. These basically regulates the operon. These are basically involved in the regulation of the operon. But if I talk of this gene, that is your leader, this leader sequence basically comprises of 2 parts. It is having another part. If you break down the leader sequence, it will have another part, as I will show you. This another part is your attenuator. Attenuator brings about premature termination of operon. So this is your attenuator. So attenuator brings about immature termination of your operon, whereas the other genes, that is your E, D, C, B, and A, these are your structural genes. So these basically are your structural genes, and they basically synthesizes the enzyme responsible for the production of tryptophan. These are the structural genes. So next comes the regulation. So next comes your regulation. Now tryptophan basically comprises of two important regulations. One is a coarse regulation, another is a fine regulation. So first we will move on to the coarse regulation. So we will move on to your coarse regulation, and this coarse regulation basically comprises, again I will draw the repressor, the promoter, the operator, the leader, and the structural genes, that is B, C, A, B, C, D, and E. So what happens actually over here is that I told you from the very beginning that this particular operon is having repressor, but this repressor is constitutive in nature. That means it is always being produced. So let's say your repressor is being produced. So this repressor I denote with the purple color. So this is your repressor which is always produced. But this repressor is inactive, and it is called as aporepressor. So it is called as aporepressor. Now, when there is a very no tryptophan in the cell, tryptophan has stopped producing, and there is no tryptophan at all in the cell or in the medium where the microorganism is growing, this aporepressor remains inactive. So it produces, but it will not be able to bind with that of your promoter. So suppose this is your RNA polymerase. So this is your RNA polymerase. So RNA polymerase do not feel any obstruction, and RNA polymerase can scan through the operon, through the mRNA, and through the DNA, and the mRNA is being produced. So if there is no tryptophan, repressor, which is your aporepressor, remains inactive, and it will not be able to bind with the operator, and hence RNA polymerase can move through the mRNA, through the DNA, and produce its corresponding mRNA. But if this is your repressor, operator, promoter, operator, leader, and A, B, C, D, E. So, but if there is a high concentration of tryptophan, if there is high concentration of tryptophan, what will happen? This high concentration of tryptophan will actually result, this tryptophan actually acts as a co-repressor. So already I have shown you in the last slide that there is a particular, this is your what? This is your aporepressor, which was inactive. But now the tryptophan, which is your co-repressor, will be synthesized because tryptophan is enough in your cell. So this tryptophan will basically bind with that is your aporepressor. So this tryptophan is your co-repressor. So it is your co-repressor, and once this is being formed, this is a very active conformation. The repressor becomes active. So what happens now? The repressor basically binds with that of your operator. This is your repressor, that is your co-repressor binding, aporepressor binding, followed by your co-repressor. This whole conformation is specific to your operator. So they are binding to the operator, and the RNA polymerase, which was sitting somewhere in the promoter, is getting blocked. It is getting blocked because there is a blockage in its path. So as it cannot scan the other part of the tryptophan operon, so what is happening? There is no synthesis of protein, there is no transcription, there is no translation. So ultimately there will be no synthesis of your protein, and that is we require, that we actually require, because these proteins are responsible for tryptophan synthesis. So if there is a high concentration of tryptophan, so we do not require to produce tryptophan. But if there is low concentration of tryptophan, then we require to produce the tryptophan. So that is only happening over here. So this is all about the course regulation of your tryptophan operon. Next I will move on to the fine regulation. Fine regulation. Now what is this fine regulation? Now this fine regulation is brought about by the presence of the leader sequence. In the last slide you have seen that there is a leader sequence. So let's elaborate or let's amplify the leader. I told you this leader is also having attenuator. Now this leader is having 5 segments. 1, 2, 3, 4 and 5 segments. And in the first segment, one segment, there are certain, these are the 2 tryptophan codons. So these are tryp codons which are present in the one, that is the first segment. Next segment are your 2 and 3. So this 2 and 3 have palindromic sequence, but this palindromic sequence are complementary to each other. So they can form a stem and loop, whereas 3 and 4 is again complementary in nature. So again 3 and 4 can also form a loop. But what is the difference in formation of loop between 2 and 3 and 3 and 4? Let's see. So if 2 and 3 is complementary, so it will form a stem loop structure. So it is forming a stem loop structure, that is your 2 and that is your 3. And this particular loop is called as anti-termination. Anti-termination means it will stop the termination. That means what will happen, the ribosome can easily move through this particular stem loop and it will produce the protein. But if I look at the other end, that is, this is your 1, 2, 3, 4 and 5. So if there is a formation of loop between 3 and 4, so this is your 3 and 4, 3 and 4, then this is your terminator or your attenuator, because it will bring about premature termination of the operon. So now we will discuss about the condition where there will be attenuation, a condition where there will be anti-termination. So let's start with that. So first I draw the leader sequence, and in the leader sequence you know that there are 2 sides, and these sides are basically your tryptophan codon, 2 tryptophan codons, and this is your 2, 3, 4, 5. Now when there is low concentration of tryptophan, when there is low concentration of the tryptophan, what will happen, the ribosome will actually bind to the tryptophan codons and will wait over there, stall over there in search of tRNA with the tryptophan. And you know that the concentration of tryptophan inside the cell is very low. So it will take time for the tRNA to come and bind with that of your tryp codon. So this particular ribosome will stall, will stop. So what will happen? It will result in the formation, formation of your stem and loop. So you get to see that you will have a stem loop structure of 2 and 3, because 2 and 3 is free. So this stem loop structure is much more favorable than 3, 4. So what will happen? This is a anti-termination loop. Anti-termination loop. So it is an anti-termination loop. So ribosome can easily passage through 2 and 3 and will bring about the transcription and translation of your tryptophan. So low concentration of tryptophan will continue to produce more and more amount of tryptophan. But when there is a very high concentration of tryptophan, this is 1, 2, 3, 4, 5 with 2 tryptocodons. So when there is a very high concentration of tryptophan, what will happen? RNA polymerase will not be able to wait. It will not wait. And what will happen? It will incorporate the tRNA over here. So what will happen? It will move from 1 to 2. So as it will move from 1 to 2, 2 and 3 will not be forming same loop, rather than 3 and 4. This 3 and 4 will form same loop, and you get to see the RNA polymerase somewhere in the 2 moving from 1 to 2. So 3 and 4 is forming the loop, and this is called as your termination loop. It is a termination loop or attenuator, and hence it will stop the transcription of your tryptophan, and what will happen? Tryptophan will not produce. So in a low concentration of tryptophan, cell requires tryptophan for survival. So tryptophan is produced due to anti-termination. But in high concentration of tryptophan, the stem loop structure is formed between 3 and 4, which is terminator or attenuator, bring about premature termination before this 5, section 5, that is your structural gene, is reached, before the RNA polymerase, the ribosome moves to that of your structural gene.